Well, welcome. As Rebecca was saying, I've been working with GAND since about 2015, maybe a little earlier than that, and it really most of the motivation for everything that's been accomplished has been through parents helping out and helping me and me helping the parents and stuff. So. It's a very important thing, and as she mentioned, we met in Cincinnati in I think 2022, and that was I think our third Helping Hands for GAND meeting in the United States, and it was obvious to me there was 2 different medical systems and probably 20 different medical systems that are being involved, and so I try my best to provide information that you guys can then go back to your doctors. I don't know if, in the United States, we have geneticists and neurologists who'll sit down with you for an hour and a half, and then we have ones that will sit down with you for 15 minutes. And it's a lot of the times you needing to have things explained to you in a nice, smooth, hopefully smooth manner and stuff, and that's what today's about. It may end up sounding a lot like a biology 101 college course, but what I'm hoping is that the information that's been provided to you, you can understand it a little bit more. Might not change much with this conversation, and perhaps tomorrow we'll give you some insight into what we're doing in the laboratory. But just wanted to start off by saying that. So, this was the first talk I ever gave at the first meeting in the United States, Understanding GAND. Basically, this was the first patient I was introduced to. The family and I are very close. They live just north of LA near Santa Barbara, and she was 20 months old at the time. I think she's 9 now. She had infantile hypotonia, motor delays, apraxia of speech. She had macrocephaly and kind of a large forehead, and kind of a distinct facial appearance. Very pretty girl. She also had bicuspid aortic valve stenosis, which required a cardiac surgery when she was young. At the time, this was not something that I would be able to recognize, and so we went on a pursuit to try and figure out what was going on. And she had an MRI, which I'm assuming a lot of this is going to sound very familiar for all of you guys, but she had an MRI that showed some extra-axial fluid collections. That's the fluid in between the skull and the brain, large ventricles, and then, in the beginning she had what was called a thin corpus callosum. Basically, the corpus callosum is just a bundle of wires from one side of the brain to the other. Again, none of this provided a specific answer. And so she had a clinical diagnosis, motor delay, language delay, hypotonia, macrocephaly, and all these things. And so the differential diagnosis was very wide. None of these features are specific to a disorder. And what we'll see later on is that there's a lot of overlap between GAND presentations and some other disorders. We did exome sequencing, which maybe a lot of you have had done, and we returned a GATA-D2B variant. On her report, it said a de novo truncating nonsense variant, and then it had these numbers and letters, and that's kind of the first part, what I want to explain to you and all. The first thing to know is de novo means that typically when they're doing this kind of testing, they're collecting mother and father and the child. And if the mother and father's samples don't have the variant, but the child has the variant, they call that de novo, from Latin, meaning new. And all that means is that the variant either arose in the embryo, when the child was an embryo, or it arose in the testis or in the ovary before the child was conceived. So to understand some of this stuff, I'm going to go through some basic DNA principles. DNA is this double-stranded helix with a code that is a bunch of nucleosides, which are just the letter, we use the acronyms A, T, G, and C, as you can see here. Now this strand is very long, and the cell, to organize it, wraps it around these proteins called histones, which when you do that, you form a nucleosome. Okay, and you may say, why do I need to know that? You need to know this to understand the later parts. And then these nucleosomes can either be tightly packed into a chromosome when the cell's dividing, or a little bit more looser packed when the cell is doing its everyday routines and stuff. If we were to take one of these strands here, what we'd see is there's a number of genes that are lined up along it. The first thing you'll notice is there's a lot of empty space in between the genes. It's even bigger than what I've written here, but you know, you can have gene A and gene B, gene C and gene D, and each one of these will have what's called a promoter represented by this bent arrow. And those promoters are sequences that unlock expression of this gene in a particular tissue. So gene C, oops, what did I just do? Okay, all right. Gene C has a brain-specific promoter, where gene D is open in all cells, and gene B would be open in heart but closed in the brain and such. Now, when you're looking at genes like this, you know, when a gene is expressed in a particular cell, and we're going to take gene C, what happens is the promoter in a brain cell will cause the DNA to be read. Now, in the form of DNA, you have coding regions, which are these blue boxes, which are exons, and you have intervening sequences, which are these black lines, which are introns. And basically, I always try and use the analogy that you're building a building. And if you're building a building, the architects or the engineers have a design and a blueprint at their office. And that's what the DNA is. And what they need to do then is to make working copies of the blueprints, which is the messenger RNA. Hello. I think somebody's trying to figure out what's going on here. And the messenger RNA is the working blueprint. And the working blueprint, if you notice, the intervening sequences here have been removed. And so you only have the coding area. And then finally, what happens is the messenger RNA is translated into protein. Now, the messenger RNA, I am not used to using this computer, so forgive me if we, I might just use my finger at that might be a little more safe. So basically you have DNA, which is transcribed in the messenger RNA, and they have the same code except for one letter's different. And then this code here is translated into protein. Now the proteins have 20 different amino acids, and there's the code in here will encode the order of these different amino acids. Now, I didn't want to say all the different ones, so I'm just using different symbols and all. But you can imagine every gene has a different pattern of these amino acids that makes the protein unique. And that's the building you're building, okay? And then those proteins can wrap themselves up into different structures. This is a hemoglobin molecule, which then builds a complex with a bunch of other hemoglobin molecules to build the actual hemoglobin we're using. So in any case, that's kind of how every cell works and things like that. Now, many of you may recognize these words because this is how you were tested. And you know, SNP arrays, they're kind of falling out of favor a little bit, but SNP arrays were good for looking at deletions and duplications, so structural changes to the DNA. So if you have a piece of DNA that's been lost or a piece that's been duplicated or things like that, the SNP arrays were very good for that. They don't use them so much because this technology is kind of caught up, and we'll get to that in a second. Some of you were diagnosed with what's called a gene panel, which is basically a collection of about 50 to 100 genes that are associated with a disorder like intellectual disability or muscular dystrophy or something like that. These are just sequence patterns that are for the different genes, and they'll also look for duplications and deletions like they do with the SNP array. These are quicker tests that in 2015 didn't exist because they hadn't quite logged GAND as a particular disease yet. Exome sequencing is a method where they sequence all the exons. Remember, that's that working part of the gene, and that's only about 1% of your total genome. But it's the meat of your genome. 80% of the issues that happen genetically are happening in the exome, which, like I said, is 1% of the total genome. Now, these days, some people may have been getting genome sequencing, which is where they sequence 100% of the genome. And people think, oh, they've got all this information, but in reality, we only have knowledge to go and cover the same things that we're covering with the exome sequencing. But the advantage of this is we still have that other 98, 99% of the DNA sequence, so that in the future we can go back and look at that. Anybody here have genome sequencing done? Okay, it's not so common, even in the United States and all, because the insurance companies in the United States are still saying, you're probably going to catch it with exome, why don't you do that? But it's catching on. As I said, exome sequencing, you're looking at the exons, which are 1% of the genome. Genome sequencing, you're looking at all of it. As I mentioned, it's becoming more and more useful. Now, both of them are becoming better at looking at deletions and duplications. So these days, the previous algorithm was you first sent a SNP array to see if there was any obvious big changes, because that was a quick test to do. And then you sent exome sequencing to flesh out if that didn't answer any questions. Nowadays, we just either send exome sequencing or gene panel or genome sequencing because the SNP arrays aren't so valuable. So, any case, hopefully now you know a little bit more about DNA, messenger RNA, proteins. You know a little bit more about the genetic testing. So back to our little girl, she had this de novo truncating nonsense variant. And then I think all of you may have seen something like this, where you have the letter C and a number, and then C to T or A to G, and then you have down here a P, and then some letters and a number and another letter. So the C here stands for cDNA, which is the equivalent for messenger RNA. I don't know why they decided to do it that way to make things complicated, but this is the sequence in that messenger RNA, and then this is what happens when the variant in the messenger RNA gets translated into the protein. And I'm going to give you an example. I'm going to give you a bunch of examples. In any case, so with this girl, she had C610, C to T. So basically, here's a messenger RNA, and you'll see here I've gone in a little bit more detail where I have the letters kind of spelled out. Now, realize every 3 letters encodes the amino acid that will be put in the chain. So that's the big trick here, is every 3, and then the next 3, and then the next 3, and then the next 3. And so you can see it's dependent on the phase here. So if you have any changes in this, some things can happen. So nonsense variants are kind of the most easy to explain, in that you're making the chain, and in the messenger RNA, there's a sequence that says, hey, here's the end of the chain. Okay, makes sense, because otherwise it might just keep going on. Now, if you have a variant, say you have this U that's going to be substituted in for the C, what happens now is that UAA is different than CAA, and instead of encoding a star here, it encodes a stop codon. And so that truncates the protein. And that's simply called a nonsense because it takes away the rest of it. I don't quite understand the logic behind why they said nonsense, but that's what it means. And so if any of your children are diagnosed where they say, PGLN204X, basically what they're meaning is that at nucleotide 610, which encodes amino acid 204, which you're going to see that this is usually a third of this, because you need 3 of these to make 1 of those. The X here stands for stop. Okay, so in this particular situation, she had a glutamine at amino acid 204 that was changed to a stop codon. And so her diagnosis is a truncating nonsense mutation that causes the disorder. And typically, if you have something like this, in a disorder like GAND, where all you need to do is have a variant in 1 of the copies of the gene, that answers the question. And I'm sure there's at least 2 or 3 of you that have something like this. Now, the next thing I want to talk about is what are called truncating frame shift variants, okay? So now you have a different situation. You have a nucleoside at 833 that's deleted, and that causes a change in the protein. So let's go here. So let's pretend this is 833, and we're going to change that T to a deletion. So basically now we've removed, that T has been lost, and now everything is going to shift over 1. Okay? And when it shifts over 1, now these 3 codon codes become different. And then usually what happens is there's a stop codon that gets put in early again. And so if you're looking at your report, you may see something like this where it says P and the G here is for glycine. So glycine 278 was changed from a glycine to an alanine. And then they added 4 amino acids, FS, frame shift 4, and then stop. So that's what that means. And so the idea here is that if you made this protein, you'd make it, and then this part would be different, and then it would be shortened. Okay? So typically, when we're looking at disorders, you can kind of use the analogy of a building again. So say you're making a building and you have a pile of bricks. Now, it could be that if you only have 50% of the bricks, when you go to build the building, it's not going to have enough to get completed. The alternative is you could have enough bricks, but maybe half of them are dysfunctional. So you can build the whole building, but the building's not stable. And so autosomal dominant disorders are like that, where autosomal dominant disorders are where you have 1 copy of the 2 copies the child has that's been altered. All right? So in a typical situation, you make the protein, 1 from mom's copy, 1 from dad's copy, and you make a normal dose. Now, what could happen is you make this truncated protein and it doesn't work, and so now you only have 1 copy, and so you have a 50% dose. Another thing that could happen is you could make the protein that's truncated, but now it somehow interacts with this normal protein, and it actually decreases the value or the activity of the normal protein, so you have a less than 50% dose, okay? And then finally, another alternative is that somehow the cell senses that this other protein isn't right, and it's never made or it's degraded right away. And so you don't, the mutant proteins you don't even see, but you do see the normal one, but you still have a 50% dose. And you might see in your reports the word haploinsufficient. So haplo means half, and insufficient means 50% dose. So half a dose, okay? Now, what we were suspecting when we were doing this was that this last situation was going on, because there's a thing called nonsense-mediated decay. You don't need to look at any of this. The important part is that the copy of the messenger RNA from the mutant version is somehow recognized by the cell and says, this is wrong, and it destroys it. And so we thought that was going on. We took some cells from some patients, and we looked at how much messenger RNA they were making and how much protein. You don't need to know how we do this and all, but when we scan these things and come up with a graph, basically this is a typical children, and then the GAN kids have about these, with the frame shift and nonsense, they make about 50% of the messenger RNA and 50% of the protein. So basically, this nonsense-mediated decay is what goes on with most kids who have nonsense and frame shift mutations in GAN, okay? And so the idea is this, what we call this LOF here, you're going to see a little bit, this means loss of function. That's kind of the generic term for nonsense and frame shift variants, okay? So loss of function variants, it's haploinsufficiency, or they have a 50% dose. And when we looked at our first study, which I'll get into a little bit more in more detail later on, if you look here, all the ones in black are kids who have variants that are loss of function, nonsense, and frame shift, which you can see is the predominant amount of, and the most common that we see with GAND. Now in green and blue, there's some other less common ones. And the next type of variant I'm going to talk about are called splice site variants. And I'm sure there's a few of you here, I know there's a few of you here, that have this type of variant. Now, this gets a little bit complicated, but again, this goes back to that first slide where I was showing you the principles of the DNA. So when you make this messenger RNA, you got to cut out these pieces, these introns. And the way the cell does that is they have unique sequences at the beginning and end of every exon. So what a splice site variant is where you have a change in these unique sequences. Now, the first thing to realize is that these splice site kind of code sequences are not set in stone. There's variability in them and stuff. And so because of that, splice site variants are a little more unique. So this is, you make what we call the pre-messenger RNA, you cut out the introns, which are represented by these little guys, and now you have the full messenger RNA. Now, if you have, so here I've blown up these sequences so that they're in front and behind each exon. Realize that when they're doing this, the cDNA is only going to be numbered where the exon is. So this could be C240 to 260, C260 to 290. This area doesn't get a number. And so if you have a variant in these things, they usually count backwards from the beginning and then give a -2. Now, alternatively, if you're going from the other side, they count forward and they give it a +2. So typically, if you have a splice site variant, you're going to see in the report that it says, A to G -2 or A to G -5 or something like that. So that's where that comes from. And again, it's when you're reading this kind of without ever seeing it before, it makes no sense. Hopefully it makes a little more sense now. Now, what ends up happening is this is my cartoon. So basically this sequence knows to go over to there, which if there's a mutant here, it ends up skipping that and it goes to the next one it recognizes. And so what ends up happening is when it makes the messenger RNA, exon 5 is absent. And when exon 5 is absent, you typically get a frame shift change again. Now, the thing about splice site variants are that sometimes, even with the mutant, it works. Because I already told you there's a little variability and wiggle room going on. So depending on how bad the change is, you can get different ratios of normal to abnormal messenger RNAs. And this makes a difference because in this particular example I'm using, 5% gets made correctly and 45% doesn't. And that's from one copy of one of the parents' DNA. So if you would put this together, the typical copy will produce 50%, like I was saying, but now you've got 5 coming from the mutant copy. So this particular child has about 55% activity, not 50. So they're a little bit above. Now that can change to as much as 20%. And so that child, so 20% gets done well and 30% is done incorrectly. And so you get about 70% of the amount of messenger RNA, which means you get 70% of the protein. Now remember, what I was saying with the nonsense and frame shifts is they're going to get 50%. So what we kind of guesstimate is that maybe the splice site kids, some of those kids might have a little bit more abilities or are on the more mild spectrum of the disorder. We're doing a study now to see if that's true or not. But that's kind of where that comes from. I know when we have the American groups together, there's a couple of kids, including one who can actually speak pretty well, and he's a splice site variant, who I'm guessing has a pretty high ratio of the good ones being made. And again, the bad copy goes through nonsense-mediated decay. So the last thing you guys may see is what's called a missense variant. Okay, now this is a little bit different in that when you have this change, which I'm going to illustrate here, that changes the code here, but instead of changing it to a stop codon, it just changes it to a different amino acid. So basically, the whole chain is being made, and so it's not a truncating one, but there's a change here that could or might not affect the ability of the protein to function. So again, here's the scenarios with a missense. The protein's made, there's a change here, and it alters the protein so that it has no function. You get a 50% dose. That's one scenario. Another scenario is that this change somehow can alter the ability of the normal copy to function, and so you get a less than 50% dose. And when we did a couple of children with missense studies, what we found was that it didn't go through nonsense-mediated decay, so there was the same amount of messenger RNA as the typical cells, and there was also the same amount of protein as the typical cells. And so we kind of look at the scenario that happens is something like this, where it's made, but it doesn't function. And we're going to get into that in a little bit more detail a little later on. So, back when our patient in Los Angeles was diagnosed with this, there was one paper out. So this came out from Holland in 2013, and it's basically a report of 4 children who had intellectual disability, hypotonia, limited speech, and a facial appearance that was very similar. Of course, because it was coming out of Holland, all of them had blonde hair, so they also thought blonde hair was part of it, and there were a few other things that they were saying there. This was then logged in OMIM, which is OMIM, which is our database that any of us can log into, and they called the disorder autosomal dominant mental retardation type 18. When I went over this, so what happened then was the family went home and they went looking for other families, and I went home and I went looking for research, and I found this, and there was very little else out there. And so as a researcher, I said to myself, this might be a good disorder for me to get involved with, because I'll get into what GATA-D2B does later, but it was gene regulation, which is what my training was. And the next thing I know, the family calls me up and says, hey, there's about 5 families here in the States. We found them on Facebook. They're really interested in getting somebody to do research. And I was like, oh, well, I'm really interested in doing research. And the Dutch group, they were basically a group that just finds new genes. So they were already on the next 10 genes that they were studying. So So basically, we started work together. The first thing we said is the name has to change. So, of course, whenever you're making up a name for a disorder, you want something that rhymes. And so we did GAD-DTB associated neurodevelopmental disorder. The rhyme came in handy for the parent group there called Helping Hands for GAND. And the next thing we wanted to do was a study just to see what's going on with these kids. Were those 4 Dutch kids really typical of what happens with GAND, or was there something else going on? We had 50 of the 72 identified subjects around the world at that time. They were in Australia, Brazil, the Emirates and such. And what we wanted to do was look at their genetics, how they presented, and their family history and stuff. And the first thing we found was their genetics, and I already showed you this, and this goes to show again those loss of function mutations were predominant. But then the green here are splice site variants, and so there was about 8 or 9 of those. And then the blue are the missense variants. And the one thing you're going to notice here is, you know, the splice site and the nonsense are kind of haphazard across the gene, but the missense variants were kind of lined up with this CR1 and CR2 region. The CR stands for conserved region, which means that when you look at this gene in humans, primates, chicken, dog, amphibians, lizards, and stuff, the sequences are very similar here. And this is conserved region 2. And again, when you go through evolution, these sequences are very similar. So that tells you that they're important, because if you change them, the thing doesn't do what it's supposed to do anymore. So the first thing we thought was, okay, the missense changes are here, so maybe this is a part of the structure of the protein that is important for its function. Oh, and I'll get to that in a little bit. So, you know, basically, when we looked at this before, about 60% were loss of function, 18 were missense, 15 splice site, and 6 deletions. And I will say, this is a little skewed because we wanted to include more of these missense and splice sites than what we were seeing randomly. If we were really doing it like appropriately, we would have done the first patient through the 50th patient. But we actually had these 70 patients, and so we said, okay, well, let's add a few of these other ones just so we can document it. The other thing that we looked at was their clinical presentations. And as you can see in gray here, this is what the Dutch group had shown. And what we found was about 88% of the kids had macrocephaly. We also showed that a lot of these kiddos had what we call oromechanical dysfunction, which means in utero, they had about 40% of them had polyhydramnios. So that's what happens is the baby's in the amniotic sac, they're urinating into the sac, they're swallowing the fluid, and then the fluid is being filtered out through the placenta in the mom. But if they're not swallowing the fluid builds up, and so they have extra fluid, so polyhydro, more fluid. When they were born, they had feeding issues, and then 100% of them, when they were older, had apraxia of speech. About 24% of them had seizures, and about 60% had abnormal MRIs, but none of the MRIs were, you know, dramatically abnormal, like what we would think of as a brain malformation or anything. It was always minor little changes. And then 10% of them had this bicuspid aortic valve, which means normally your aortic valve, which is controlling flow out of your heart, is shaped with 3 valves like a peace sign. And instead, they just have 2 valves, and when the heart beats, it opens up. Now, in the general population, about 2% of us have this. And the only issue with this is that with just 2 leaflets, what can happen is it can calcify at the 2 corners and cause that to get smaller and smaller. And if you remember, my first patient, it had calcified so much that they actually had to go in with an angiogram and put a balloon in there and blow it up to open it up more. When it came to their facial features, we saw that a lot of them had high, wide foreheads and what we call frontal bossing. Many of them have wide set eyes. Their nose typically looks very similar. They tend to have a very short philtrum between their nose and their lip. And they also have pointed chins. And you know, the interesting thing is, the first time I was at a Helping Hands for GAND meeting, I walked into a room and there was about 10 kids, and I was like, well, either these are all brothers and sisters or they're all kids with GAND. And I think if you look around the room, you'll notice they all very, very much look like they're siblings. When we look at the data, I already kind of went through a lot of this. You know, many of them are nonverbal, less than 4 years. About 20% are nonverbal above 4 years. When I say nonverbal, that means no language at all. Many of them have a wide-based gait, meaning they're a little bit unsteady on their feet. Many will have orthotics, like either ankle braces or foot braces, and only one had deafness. And I'll get into why I looked at deafness a little bit later on. And by the way, anybody who wants this paper, I can send it to you, or I can send it to Rebecca and stuff. But it basically has all these different parameters we went through. You know, less than 4 years, almost none of them were toilet trained. And even above 4 years, you know, only about 30 to 40% were toilet trained. That tends to be one of the big things that families are asking about. As I said, a bunch of them had bicuspid aortic valves, and 2 of them had to have this kind of heart surgery. I think many of you know that a lot of these children have strabismus, or I think you call it a squint here, and either need to have surgery or wear glasses. They also can have a couple other things like astigmatism, and those were the other features I've already gone through. So nowadays we've got about over 450 cases identified around the world in all. And what we're doing now is we're having a new study where we want to get a little bit more of these kids with missense splice site intragenic deletions. That means that there's a deletion of the DNA within the gene, and then extragenic deletions where there's a deletion that's the gene is involved, but the deletion's even bigger than the gene. This is ongoing, and I just lost my train of thought for a second, so forgive me. We're also looking at parental mosaicism, which I'm going to talk about a little bit more tomorrow. And then in the future, we're hoping to do a neuroimaging study. So if any of you guys have had MRIs done and you can get a disc of the images, please collect them from the university or the hospital and just keep them, and then probably within the next year or 2, I'm gonna start a study for that. So, some things to know about GAND and GATA-D2B. I just have to take a little bit of a break. So I'm going to go a little bit on the nomenclature. And this may be a little confusing, again, when you're reading things and you're like, why did they write it this way and why did they write it that way? Just some basic housekeeping. A human gene is going to be in all capital letters and italicized. A different species will have its own unique things, but say a mouse has a capital letter, lowercase, and italicized. The protein is going to be capitalized, but not italicized. And like in a mouse, it's going to be written where it's only the first letter is capitalized and stuff. That's not that important, but I'm sure if you're reading things through, you're going to be like, why are they doing that? So, what does GATA-D2B do? Well, GATA-D2B is a part of a complex called the NERD complex. And the NERD complex comes, I didn't name it, is the nucleosome remodeling and deacetylase complex. I'm sure almost none of you know what that means, but in any case, you're going to know in about 5-10 minutes. So basically what this is a complex of different proteins. Some of them are 2 copies, like HDAC and MTA. Some of them are 4 copies, but these 3 are only a single copy, okay? And GATA-D2B kind of links these guys together. This is the histone deacetylase core. We're not going to talk about that. We're going to talk about this nucleosome remodeling more. So if you remember this slide, you might remember how I said, why are you learning this? Well, again, you have your strand of DNA, you have these protein histones that organize the DNA, and when you have the protein and DNA together, that's a nucleosome, okay? And again, GATA-D2B is part of this nucleosome remodeling and deacetylase complex. Now, my collaborator, Joel Mackey in Sydney, Australia, he put together this 3D image of what they figured out. If you look here, you can see there's a piece of DNA right there, and then here is the histone protein. Now, NERD has that big green protein, and then the gold protein here is GATA-D2B. And then there's another, this is that HDAC complex there. And so it's interacting with the DNA and nucleosome through the HDAC and through this big green protein. And if you look, GATA-D2B is the bridge between the two of them. So you need GATA-D2B to have the whole complex come together. Now, what they knew about the NERD complex back when I was starting is that it's involved in stem cell differentiation, genesis of neurons in the brain, and then the development of the cerebral cortex, which is the part of the brain that we're all kind of using right now to listen and hear and stuff like that. And as I said, you have this blue histone deacetylase core that we're not going to talk about, the gold GATA-D2B, and then the green CHD protein, but that's a nucleosome remodeling protein. So you need to have both of these activities together for NERD to function, and GATA-D2B holds them together. Now, remember I was saying how CR1 and CR2 are conserved across species, and that we noticed that these missense variants were located within those. Well, basically, the missense variants are where in CR1 are where the HDAC core and GATA-D2B go together, where in CR2 it's where this green protein and GATA-D2B come together. So that's why you can have a little change of the structure, and these guys are kind of like Legos, and if you imagine you change the pattern of the Legos, they're not going to fit together. And if they don't fit together, they're not going to form the whole structure. And so that activity as a bridge is lost. And so, CR1's interacting with MDD3, that gets lost, and CR2's acting with CHD3 or some of the other proteins there, and that gets lost. So that's how a missense variant happens. And when we were initially doing this study, I saw these missense variants, and I called Joe Mackay in Sydney out of the blue, and I said, hey, I'm working on this protein, I see you're looking at the structure of it, would you be interested in modeling some variants? And he was like, oh, I've been waiting for this phone call for a long time. And within a month, he had a graduate student who modeled the variants and showed that the green protein doesn't interact with GATA-D2B because of the missense variants. And so that was us using patient data to make a hypothesis that we then tested in the laboratory, and it came true. So that's why doing these clinical studies are very valuable, because we can start making kind of suppositions to go, okay, what's next? So, any case, if you thought that was complicated, it gets more complicated. So, the first thing to realize is that at every one of these sites, there's more than one version of the protein. So, where the green site, there's a protein called CHD3, there's one called CHD4, there's one called CHD5. So if CHD5 is here, then the other two aren't. If CHD3 is here, the other two aren't. There's actually another GATA-D2 protein called GATA-D2A, and such, and all of these spots are like that. And this is why when I saw that patient the first time and I looked up the research, there wasn't much data out there, because this is very hard to figure out how to study. Because any effect you can see, someone can say, well, it's because you're not looking at the right protein here. So again, The NERD complex is actually a bunch of different NERD complexes. You can have some with CHD4 and GATA-D2B. You can have some with CHD5 and GATA-D2A, and stuff like that. So it makes it very tricky. But one of the things we did notice was that when we published our paper on GAND, these sort of symptoms were seen, and then we had our other unique symptoms with GAND. And again, this is NERD here with GATA-D2B. Well, interestingly enough, there was another paper out on a disorder related to the CHD4 protein. And they had very similar presentations. They had heart defects that were a little bit more severe, but then they also had a couple of things that weren't very common in GAND. They also had their own unique facial appearance. Then another paper came out about CHD5, and again, this is NERD with CHD5 and GATA-D2B. They didn't report that the kids had macrocephaly, but I'm pretty sure they do. And they had very similar features as well. Another one came out with CHD3, where they have macrocephaly, intellectual disability, motor delays, apraxia of speech. They also have male genital anomalies, which isn't seen in GAND as yet. That's part of the new study as well. So when we saw this, we started kind of trying to put this all together. And so if we look here, this really should say GAND. Here's all the features of GAND, or some of the big ones. And if we look at CHD4 related syndrome, you have overlap along with the heart defects, but these two things are not really overlapping. If we add CHD5, they tend to have a little bit more epilepsy and such. And then if we add CHD3, again, the overlap is with apraxia of speech. Now, I did this so that you could imagine this is the rest of NERD, GATA-D2B, and CHD4. And then the rest of NERD, GATA-D2B, CHD5, and CHD3. So basically, if you're going to have CHD4 work, it needs a GATA-D2 protein to connect it to the rest of NERD. The same with CHD5 and CHD3. And so to me, I kind of realized, like, hey, these are all kind of related to neurodevelopment. So maybe GATA-D2B is important in neurodevelopment, where GATA-D2A is important earlier on, okay? And so maybe that's involved with setting up the heart a little bit more, because that happens before the brain. And maybe that's why CHD4 has a little bit stronger heart defects, because maybe GATA-D2A and B are working with it there and stuff. So basically, I came up with this idea called nerdopathies. And what I really wanted to emphasize is that GATA-D2B and GAND is kind of all of these disorders together, except for a few symptoms. And the idea would be that when the brain is being developed, if you don't have a lot of GATA-D2B, say you have 50%, you're only going to have 50% of these guys, okay? And this was kind of the diagram that finally got me over the hump when it came to getting grant awards, because just like I was saying earlier, everybody who would read my grant things were saying this is too complicated. And once I said, listen, this is actually the key to all the NERD activity when it comes to brain development, they were like, oh, now you have a model that you can move forward with. I hope everybody's following me there. So again, we're doing another study with this stuff, and the idea is that, say with the missense, if we come up with new spots where there's missense variants, that might tell us new areas of importance in the gene and stuff. Now, interestingly, one of the things that we're doing is we're also looking at what I call NERD-related disorders, meaning disorders that either affect NERD or also affect other proteins that interact with NERD. So, like SAPB2-associated syndrome is related to a protein called SAPB2, which is involved in brain development that NERD interacts with to program cells in the brain. And if you look at the symptoms of SAPB2, again, you have a lot of the same issues. You know, I haven't seen a lot of children with GAND who've had any real severe dental or palatal changes, but I'm on the lookout for that because I'm thinking maybe this is one of the big interactions with NERD that sets off the disorder, because the more similar these things are, the more it says they're related to each other. Now, one of the big things we see with SAPB2 is apraxia of speech as well. And if I'm going to go back again, apraxia of speech is also seen with CHD3. So, you know, when we're doing our research, one of the things we found is that there's a protein or two proteins called FOXP1 or FOXP2 which interact with CHD3 and GATA-D2B. And this disorder can cause a pure apraxia of speech, meaning the children can have normal intellect, but they just can't vocalize. And so one of the things that we're thinking of is that there's perhaps a cell type or a group of cells that express CHD3, FOXP2, GATA-D2B, and SAPB2 together that lead to expression of being able to speak enough. So this is just kind of a basic primer for what we're doing. It sets up a little bit about what I'm going to be talking about tomorrow. But one of the big things that we were realizing is that SAPB2 here is a big player in the development of the cerebral cortex. And the fact that NERD and SAPB2 work together in that process kind of point us in some directions, which I'm going to talk about a little bit more tomorrow. Now, I want to just give some acknowledgments. I have to apologize to Rebecca. I didn't put in GAND UK in my acknowledgments because I was a little jet lagged last night and I forgot to do it. But this is the family in Los Angeles. You know, I'm still friends with these guys. Last year, she got to get on the football field for the Los Angeles Rams and be like the captain. This is my team. They don't like their picture taken, but this is Halloween. Gabby Otero is one of the researchers. This is me, and this is J-Min. I get a lot of my funding from the Fashion Industries Guild, which is a philanthropic group at Cedars, as well as getting some help from the Svensson and Brunig lab there. I know you guys probably have a lot of questions, and I think we have time. I don't know where Rebecca is, but I think we have some time for them. So can I answer any particular ones? Well, thank you for your attention, by the way. Don't be shy. Go ahead.
Earlier in your research, you had some numbers about toilet training. How do you define toilet training being done? For example, our daughter, she's 4, she's turning 5 in October, and we were able to be done with daytime toilet training when she was around 3.5, and she still wears a diaper at night, which sometimes, you know, she has a little accident in the morning, but other than that, she doesn't wear a diaper anymore. So
yeah.
How do you define toilet training?
Well, in general, it's their ability to communicate the need to use the toilet and stuff, and not just go without knowing. So, in general, it's a loosely defined concept and all. And, you know, the more people that we're getting information from, the more we're learning that some of the kids are doing a lot better than what we were initially reporting. The first thing you guys should always realize when it comes to any sort of clinical study or genetic work is the most severe kids tend to be getting the most work done and the fastest work done. And so there's always a little bit of a skew towards the more severe side in the beginning. And then later on, the numbers kind of settle out a little bit over time. So if, you know, once we're done with the missense splice site cohort, we'll probably try and do another cohort where we're just randomly enrolling people just to see where the numbers fall out now that you know, people are getting genetic testing much more than they were before and stuff like that. Any other questions? Of course, you guys can always come up to me afterwards and ask me specific questions about your child and stuff like that. But I really appreciate you spending the time to listen and stuff. And like I said, tomorrow I'm going to talk about our research. It might be a little more complicated, but hopefully I'll be able to get it down to brass tacks so you understand where we're going, what we think we're seeing, and what we don't think we're seeing. Thank you so much.